So, I'm going to give a rather strange talk because it's going to be somewhat general about some ideas about how to do science or how to get ideas or what to do. And the title is a little strange. As you can see, it's the continuing need for useless knowledge. And useless knowledge is basic research. So carrying on from what Peter showed just a moment ago. But the title actually comes from a paper that was written in 1939 by this man, Abraham Flexner. Now, in the United States, people go to medical school after they go to college. And so these so-called pre-medical students have a lot of requirements they have to do. They have to take physics, they have to take chemistry, they have to take biology, they have to take math. There's a whole series of requirements. In 1910, he's responsible for that. He was the guy that has ruined the lives of pre-med students in the United States for over 100 years because they're still following those instructions. But he also happened to be the person that started the wonderful think tank institution, the Princeton Institute for Advanced Study, where people could come and just think. So Einstein and Gödel and just an enormous number of mathematicians and physicists have benefited from being able to have a place they can go and think, talk with colleagues, develop new ideas. But in 1939, he wrote this article, The Usefulness of Useless Knowledge. And as you can see here, it says, intellectual life is on the surface a useless form of activity, but it is unexpectedly the source from which undreamed of utility is derived. Now, he starts the article off by saying that he had a conversation with the industrialist George Eastman, that person who started the Eastman Kodak Film Company. And he's talking to him and he says, George, who do you think is the most important scientist in the world? And Eastman says, that's an easy question, 1930. The most important scientist is Marconi, because Marconi developed the radio and wireless. Clearly very important inventions. And Flexner in the article basically says, I told him he was nuts. That in fact, Marconi was just inevitable. Somebody else would have done it. What was really important for the development of radio and wireless was an understanding of electromagnetic waves. And that came about from the work of Clerk Maxwell and Hertz, because they were the ones that came up with the basic understanding of electromagnetic waves. And without them, nothing would have happened. And then he goes on to talk about other things, and I'm not really going to talk about it here. But I want to point out that I think, to one extent, he is right. I think you need new knowledge. You need basic research, but you also need to apply that. So I'm not talking that it's one or the other. I'm talking that it's both. The need for new knowledge that allows for new applications, and both are really needed. So I'm going to give a couple more examples of this idea. Also from something even earlier than 1939, a letter that the writer Samuel Clemens, otherwise known as Mark Twain, wrote to Helen Keller in 1903, in which, as you can see, he says, it takes 1,000 men to invent a telegraph or a steam engine or a phonograph or a photograph or a telephone or any other important thing, and the last man gets the credit, we forget everybody else. And to maybe bring this more up to date, I want to show you this wonderful infographic. I will warn you, you will not be able to read it. But it's an infographic from the Quartzoff company. And if you go online, you can look at this. What is this an infographic of? If we look at the very top, this is all the science that leads eventually to the iPhone, to a smartphone with its computer and its capability for telephone and wireless and computation and camera and all the other things that we have in our smartphones, what was needed for that? And as you start going down the chart, just going a little bit lower, the arrow will show you where I'm going, right down. Oh, nope, I went too far. Sorry. Let me go back, try it again. There we are. All of this was needed first before making up. And as you keep going down the chart, you find more and more things until you reach the bottom. And in the bottom, you find the first lens, the discovery of zero. All the things that were needed to allow for the mathematics, the physics, the optics, everything that are needed that eventually all culminate in this one application. So lots of things are needed for this altogether. And I have a favorite video. It was made in 2014, so it's a little bit out of date for some of the things about NIH. But it was made by these people at the bottom. These were all graduate students and postdocs at the University of California in San Francisco. And what they were asked, they were competing in a competition from the Federation of American Societies of Experimental Biology, and they said, make a video, a short video, explaining the importance of basic research. And this is what they did, and I hope the sound will come up and we will be able to hear it, and I will shut up.
It's a sunny day in San Francisco. We set up a table by the water and asked people this question. If it's the year 1960 and you had $10 to give to science, would you spend it on A, developing an affordable treatment for diabetes, or B, figuring out how bacteria protect themselves? The majority of people we surveyed wanted to spend their money on a diabetes treatment, but what really happened 50 years ago? The government decided to spend money on figuring out how bacteria protect themselves against viruses. The National Science Foundation, NSF, and the National Institutes of Health, NIH, are two government agencies that allocate tax dollars to scientists. And they decided to fund groups of researchers who thought that studying how microbes protect themselves was an interesting question. At the time, there was no way of knowing how this basic research could affect human health. They were simply curious about understanding something about bacteria biology. So what did the scientists find? Normally, when a virus enters a bacteria, it adds its own DNA to that of the bacteria. In doing so, the virus takes over the bacteria and uses it to make more viruses. Scientists found that bacteria protect themselves from viruses by selectively cutting the viral DNA. This basic finding led to a scientific revolution. Scientists learned how to cut specific chunks of DNA, and they could use this technique to move genes around. They could even move human DNA into bacteria. And because bacteria are very good at making more of themselves, scientists can use them to make large quantities of human proteins from the human gene that they introduced. So why was this technique useful for modern medicine? Well, one breakthrough was the efficient production of human insulin by introducing the human insulin gene into bacteria. This was a very significant advancement in diabetes treatment. Before using bacteria to produce large quantities of human insulin was expensive, unsafe, and less effective because it was harvested from fetal cows. In fact, the first person to get an injection of cow insulin suffered from a severe allergic reaction. Because of a long line of federally funded basic research, human insulin is now an affordable treatment for diabetes. By asking how bacteria protect themselves, the scientists discovered that bacteria can cut DNA. This discovery was developed into a basic technique used in thousands of labs around the world to create new knowledge and to ask new questions that have led to all kinds of therapies and technology, including insulin, DNA fingerprinting and biofuels, the human genome, growth hormones, cancer drugs, stroke medication, vaccines, HIV meds, antibiotics, and many, many more. Given enough time and funding, who knows what is to come? Many other health benefits and current discoveries would have been nearly impossible without federal funding for all kinds of basic research. Basic research is an investment in our future. It broadens our understanding of the world we live in, and the potential benefits are limitless. Unfortunately, science funding in the U.S. has plateaued, decreasing purchasing power and significantly hurting our competitiveness and innovation. You may not know it, but you and your tax dollars are part of the discovery process. Thanks to you, scientists discovered that bacteria can cut DNA. And thanks to you, insulin, other drugs and diagnostic tools have been developed and are helping patients.
So thank you for supporting basic research.
We are the scientists who depend on you, and we are excited for the future that lies ahead.
I'm very impressed with that. That's a wonderful, wonderful declaration. Let me go on from here to talk about another thing. In the United States, we have an award that is called the Golden Goose Award. And as you can see here, a Golden Goose Award is basically for something that sounds like it's really ridiculous, but turns out to be very useful. And fortunately, I was considered one of those ridiculous people because we, the three of us, Roger and Osama and I, won it for green fluorescent protein. But the maser, which was the precursor of the laser, also got it in that first year. And the people that discovered that coral, when cleaned up, is a spectacular substitute for human bone. And so for replacements, it's a wonderful substance. But the one I want to tell you about is some experiments that won a later version of the Golden Goose Award. And those were experiments that had to do with rat and infant massage. So there were a group of researchers, and they're doing some work with rats, and they make a genetic variant because they wanted to see what the animals were going to develop as. And they were very concerned that because these mutants might not be exactly like the normal pups, that the mothers might eat them or reject them or do something so they wouldn't live. And so they decided, when they were doing the experiment, that it would probably be a good idea to separate the pups from the mothers as soon as possible and from the other pups. And so they took them away, and to their horror, they found that that was a disaster. The pups really didn't thrive at all. So they decided maybe that wasn't a good idea. They put the babies back with the mother rats, and the babies did fine. And that led to a question. Why are the babies doing fine? What was different in the two? They had the same nutrients and everything. What was wrong? And they found, by observing the mothers, that they were constantly licking the babies. And so they decided to do an experiment. They took the pups away from the babies. They didn't lick them, but they did take a swab and sort of rub their backs. And they found that massage, that stimulation, that mechanically gated activity led to the pups thriving. But they didn't stop there. They started thinking, well, maybe that's an important thing, that contact, that massage, that's an important thing. And my understanding is it's used in every neonatal ward in hospitals all over the world now as a way of helping premature babies thrive. We don't have human mothers lick the babies, but that's okay. But you never know what the consequences of the work is going to be or what's going to come out of this. But I think that one's a very wonderful example. I think that in order to have the new knowledge, when we have new knowledge, it gives new approaches, and we have to figure out a way of getting new approaches. I think there's, over years of people talking about just what you saw in that video, that although I think it's very understandable that patient groups are very concerned about their loved ones, they want cures. But I think an argument has been made over the years that while people should be working directly on the disease, not abandoning that, we need to have more knowledge to come up with new approaches for the disease, that it's a multi-pronged activity to do this. And I think advocates for disease now see this as an important aspect of the overall work, that we need new ways of thinking and new knowledge about the human body to be able to come up with new approaches. But I wonder if this is maybe something that we should be doing more generally. What do I mean by that? I have a very good friend, Manu Prakash, who's an engineer at Stanford. If you've ever heard of him, he's the one who came up with this $1.50 microscope called the Foldscope. It's made out of paper, and he sent 2 million of them all over the world for people to play with. But now he's very much interested in climate change. And I don't know what happens here in Hungary, but in the United States, you turn on the television, if you hear anything about attempts to deal with climate change, it's always carbon sequestration or some sort of mitigation or electric vehicles or different windmills or some sort of engineering solution. And what Manu has been saying now for a couple of years is, that's good, but we are fundamentally ignorant about what is actually happening in the world. And the example he likes to give is that we really don't understand how the oceans, which are the major sink for carbon dioxide in the world, how they really work, how this works with microorganisms, what is it at the depths, because we have never really looked at such a large scale. And so it's really something new that has to be looked at, and it's the basic research. So we have to do these mitigation things, that's clear. But we also have to be thinking about what is the understanding that we actually have of the world, and that requires more investment in basic research. So how do you make discoveries? The physicist Enrico Fermi has a wonderful quote about the difference between measurements and discoveries. He says that if the results of your experiment confirm your hypothesis, you've made a measurement. But if they don't confirm your hypothesis, then you've made a discovery. So when it's the unexpected. So the question really is, how do we get at this unexpected? And I want to give a couple of examples of such unexpected discoveries that have actually resulted in a Nobel Prize. So the first one I want to give are these two guys, Bob Wilson and Arno Penzias. They were at Bell Laboratories, and Bob Wilson wanted to, he was sort of embarrassed. His physics PhD, which he got, the experiment actually didn't work, and he wanted to repeat it. And he found that there was this wonderful 30-foot microwave telescope that somebody else had made and was just sitting in the grounds of the Bell Lab, and he could use that for his experiment. So he and Arno get together, and the first thing they do, they're very good scientists, they're going to check out the machine. So they know that if they point it at a lot of stars, they're going to get a great signal. So they point it at the Milky Way, and it works beautifully. They get this very powerful signal. Then they say, okay, now we're going to zero it, and they point it away from the Milky Way, and it doesn't go to zero. They cannot get the machine to go to zero. No matter where they point it in the night sky, there is something there. They first think maybe it's a problem with electrical lines, or maybe it's airplanes flying by their installation, or maybe it's actually the pigeons that every once in a while roost into this 30-foot telescope. They get rid of all these possibilities, and finally they realize that that's real. It's not zero because there is a background radiation, which was the first real indication of evidence of the Big Bang. And that's why they won their Nobel Prize. So they never actually did an experiment to get their Nobel Prize. They were still just trying to get the equipment to work, and then it led to it. Another person is John O'Keefe, neurobiologist in London, and he discovered these really interesting cells in the brain that seem to order place, your position in space. He calls them place cells. And in his Nobel speech, he says that he was doing electrophysiology in the brain and looking at the thalamus when one day, by mistake, he put the electrode in the wrong place. And he found these really interesting cells. And they eventually led to his getting a Nobel Prize. The third example I want to give is an example where a Nobel Prize was given for two mistakes or two unexpected discoveries. And this is the work of Osamu Shimomura, who shared the prize with GFP with me. Osamu was an astonishing biochemist, very talented, and was working on a wonderful, wonderful problem. How is it that some organisms have the chemistry to generate light? We have all sorts of organisms that do that. Fireflies, glowworms, bacteria, fungi, fish, all sorts of organisms generate light. But they all do it in a different way biochemically. So he had as his project in 1962 to figure out why this jellyfish, Aquaria victoria, produced green light. And so being a good biochemist, he ground up a lot of jellyfish. Ground up so many he had to start bribing his kids to get him more. And he did, but it was a penny for 100 jellyfish. He's a hard guy. And he makes the preps, he's doing all the standard biochemistry, nothing. Goes for a whole summer, every time he tries something different, it just does not work. He cannot extract the bioluminescent molecule from the jellyfish. One day, he's working all day doing the experiment. It has failed again. And he finally, it's late at night, it's dark out, he wants to go home and have dinner. He says, okay, that's it, I failed again. He takes the whole prep and he throws it in the sink. He turns off the light and he's about ready to leave when he happens to look back at the sink, which had seawater in it, and the sink is glowing. This is the scientific method. You can throw things in the sink, throw them on the floor, on your lab bench, and then do the experiment. Sometimes it works. So he thinks about that, and he says, well, I wonder why it suddenly worked. And he thought, maybe there's something in the seawater that's important. What does seawater have that I haven't put into my prep? Calcium ions. And so a couple days later, he redoes the experiment, and sure enough, every time he squirts in a little calcium, he gets a flash of light. And he uses that to purify the protein. And when he purifies the protein, he names it after the jellyfish, and that protein is called aquarin. And so aquarin plus calcium produces light. So he solved the problem. What is the molecule that allows him to produce light? Except, I'm pretty sure he said, oh, nuts, or something else probably, because he looks at the light that's produced by this thing, and it's blue, but the jellyfish are green. So he's got the wrong color. So he adds calcium probably to all the other samples, and that doesn't work either. He doesn't have it. And then he thinks, well, maybe there's something else. So that's the second accident, the wrong color. And he goes, and he says, well, maybe there's something that converts the blue light to green. So he gets a lamp, a blue light lamp, and he starts looking at every one of his samples, and sure enough, a completely different one from the one that had aquarin has something in it, a protein as it turns out, that converts blue light to green. And he writes a paper in 1962 on the purification of a corn. And in it, he has a footnote number 3, and where he says, there's this other protein. I'm going to call it the green protein. If you shine blue light on it, you get green light on it. Of course, we call it GFP. Doesn't need calcium, doesn't need a corn. All it needed was blue light to do this. And he discovers the first fluorescent protein. As far as I know, this is the only footnote that has ever won its writer a Nobel Prize. So you don't have to write a lot. And I heard about this not in 1962 or in the 60s or in the 70s. I heard about this in 1989. Nobody was paying any attention to this, but I heard this wonderful seminar in which it was described, and I realized, because I was doing work on gene expression and I was doing work on an animal that was transparent, that if I could get the gene to turn on the making of this protein, I would be able to see where that was made simply by shining blue light on the organism. Because all you really need is blue light coming in and you get green light coming out. I want to also point out, this is an astonishing molecule. This is the structure of GFP. And it's the only molecule I know of that actually looks like it should. Because it's 11 strands making a can, a barrel. The 2 parts come in. So it's a can and 1 alpha helix going down the center. It is exactly what it's supposed to be. It's a lantern. It's quite remarkable. And so I said, we got to put this in animals. And I was very fortunate in the lab. I had a wonderful graduate student, Gia Skirkin, who had just come to the lab, who knew about fluorescence. And I had already contacted Douglas Prasher, who was in the process of isolating the DNA for that. I should tell you the color code. The color code for my talks is always the same. Anyone in my lab, like Gia, their name is in red. Anybody who I've collaborated with, their name is in blue. And if you see anyone like Shimomura, whose name is in black, they did something I wish I had done. Okay? So Gia started off, and the first thing she did was put the GFP that she got from Douglas into bacteria, and she saw green fluorescent bacteria. So we knew it worked. We then put it into worms, and then for the first time in a living animal, we could see the cells that we were studying. This gives up an enormous number of opportunities, because once you can see something, you can study it. You can mutate the animal and look for animals that have fewer cells or more cells, or cells in the wrong place, or cells that have more or fewer branches to them. All sorts of things once you can see something. And other people put this in all sorts of organisms, like flies and zebrafish and mice and plants and all sorts of bacteria, archaea, all sorts of different things. And one of the nice things about GFP is it diffuses throughout the entire cell, so you can actually see, as in this Purkinje cell, the entire architecture of the cell, the entire structure. And so lots of people used it, and it was very exciting because of all the possibilities of study and manipulation. But the most important thing about it is that it worked in living organisms. All you had to do is shine blue light on it to be able to see it. And I want to show you 2 quick movies made by Rosalind Silverman Gavrila. At the time, I think, was a graduate student in Canada. I've never met her, but she put these movies on the website for the American Society of Cell Biology. On the left, GFP is attached to a major protein tubulin of the spindle. And you're going to watch 4 rounds of nuclear division of the early fruit fly embryo. There are no cell boundaries here. They're just nuclei form and then unformed. So let's watch that. And there's the spindles forming. The division takes place. Very much sped up here so we can see 4 rounds. And the interesting thing about this, and one of the advantages of seeing something live, is that you see that it's synchronous. In fact, she named this movie In Synchrony, and it immediately gives rise to questions of what is the coordination among all of these divisions? What's happening? What's the biochemical background to controlling these divisions? Now, the image on the right, on your right, is falsely colored. And it's still just GFP, but if you have a lot of GFP, you're at the red end of the rainbow, and the convention, as you get less and less down to blue, I'm going to keep hitting that. As you go down to blue, that's less and less. So what we're talking about is the amount. So right now, and this particular construct was GFP with a small peptide called a nuclear localization signal. And so with that, all the GFP goes into the nucleus. But when the nucleus breaks down, when there's division, it's going to go all over the embryo. So let's watch that. And it breaks apart. She calls this Starry Night after the Van Gogh painting, and you'll see why in a second. Now it's everywhere in the embryo. Now it's starting to be sucked up by the new nuclei again, and then they go through another round of division. And you see here it's not synchronous. It's going from the lower right to the upper left-hand corner, and we believe that that's because maybe her thumb was a little heavy on the slide when she made this, and there's a little bit of damage. But it shows you how sensitive this is to be able to look at this. Now, GFP has been used in thousands upon thousands of experiments in a lot of different ways, and it's like any discovery can give rise to more basic information, more fundamental information. So the one I like very much is by Cliff Brangwyn and Tony Hyman, who labeled a cytoplasmic protein and looked at it in the same worm that we work on and discovered that this cytoplasmic protein wasn't dissolved equally in the cytoplasm, but it looked like it was like oil in water. These are called phase-separated particles. These are probably one of the hottest areas of cell biology today. People wonder if abnormalities in things like this might lead to the aggregation that happens in various neurological diseases and stuff. So this is a profoundly important thing. It's not only in the cytoplasm, it's also in the nucleolus, it's in other places in the nucleus. It seems to be connected sometimes with RNAs and with transcriptional regulation. There's all sorts of new things that are being discovered to this. No one had seen it before they used GFP. Now, I'm going to give you an example that's more applied. About the same time as this experiment was being done, some researchers wanted to look at what happens to the virus that caused AIDS, HIV. How is it transferred from one cell to another? If you've taken any elementary courses on viruses, usually bacterial viruses, they talk about cell lysis. The cell explodes and the virus goes everywhere. Well, if the virus goes everywhere, if it's out there, then maybe antibodies could come to it and scarf it up and get it out of the way. But what actually happens, or what did they find? So what they did is they took a mouse cell, put the virus in the mouse cell, infected the virus in the mouse cell, but the virus had GFP in it. So you could see an infected cell if it had, because of the GFP, and then they could see what happens as that virus gets transmitted to another cell. So let's see that movie. So there's the infected cell with the virus. There's a virus particle there, and you can barely make out that there's another cell there, and then all of a sudden, it's been transferred. No explosion. And so this immediately says if you want to control the transfer of virus from an infected to a non-infected cell, you really have to be thinking about other things. You really have to be considering this. So, as I say, it's been used for fundamental research, for applied research, in this case medical. But I don't want to give the impression that fundamental research always gives rise to applied. it's really a two-way street because it keeps going back and forth, and there's a third thing that comes out, really unexpected things. So let me quickly show you some unexpected things. There's a group in Japan that put GFP and other colored fluorescent proteins into silk moths to make fluorescent silk, and they made fluorescent clothing out of this. I've said many times that I'm not really sure how useful this is because you have to walk around with an ultraviolet lamp all the time over you to show people how wonderful your clothes are. But it was an interesting idea and something I hadn't expected. Other people made a cellular laser using GFP in their experiments. Bob Berledge, whoops, I'm going to fall off this way. Bob Berledge knew that two things. One, that there was a control element in bacteria that would allow for gene expression in the presence of the explosive TNT. He also knew that landmines, one of the worst aspects of war, well, they're all horrible, but they kill innocent people, non-combatants. Landmines leak TNT. And he wanted to develop a way to detect landmines. So he made bacteria that would glow green because of GFP in the presence of TNT by taking the control element and having it allow for the production of GFP. He then had a friend bury 5 landmines, disconnected, because he's not stupid, but still with TNT in them, in a 5 by 3 meter plot of land. And then he sprayed the area with the bacteria, and then he went back with a UV lamp, and he was able to see where the landmines had been buried. So this is a wonderful approach. Has nothing to do with medicine except to prevent the need for doctors, I would say. But to me, this is a great example of thinking about other applications, about other things, about other stuff that you can do. And I think it's terrific. But I will say that it's still not perfected after many years, because this is one thing you do not want to have a false negative. You want to make sure that when you don't see it, it doesn't mean that if you don't see it, you want to know you're safe. So false negatives are the problem here, and people are trying to work on that. Other things that have been done, GFP has been used in art. You just saw an example in Peter's introduction, but it was also used by the artist Eduardo Koch to make Alba, the GFP bunny that he brought to his art shows, and Ang Lee used the idea of GFP as the reason the Hulk is green. I actually know the writer for his, James Shamus, who is the screenwriter for this play. His daughter and my daughter happened to go to elementary school together, and I asked him how he knew about my research, and he said, "I don't know anything that you do." It turned out a student was working on the set, and the student suggested to him, "Why don't you give a reason for the Hulk being green?" It's GFP. So that's the start of the movie. You see a jellyfish and a hypodermic taking out the GFP. It's quite nice. Anyway, if discoveries are accidental, how do we get more? And I'm going to suggest 2 different things. One of them is that we have questions in front of us all the time that we don't consider, that are there, but we are oblivious to them. And I think we should try to address more and more of these questions. The second thing is I think we have to question our assumptions about things. And I'll give a couple of examples of this. Let me start with the question of right in front of it. And I know some people in the audience have already heard part of this talk, so you guys can't answer this question. But the new people here, my first scientific paper involved the part of the human body, or the part of the body that's transparent. So if you know what that is, raise your hand. Very few people raise their hand, and that always happens. Some people might be shy, but I will now give the rest of you a hint, and that is it's right in front of your eye, because it's the cornea. But I have never met anyone other than the person I happen to have the luck of going into his lab to study this, who ever asked themselves the question, why is this tissue transparent? How is it that I can see? What makes that tissue different? A question that's been in front of us our entire lives. So other things like this. I have a couple of other examples. I'm not going to show them for time, but I will tell you about a remarkable person. He's my cousin. Ray Goldstein is actually a second cousin. He's a professor of physics at Cambridge University in England, and he's made an entire career of answering these questions. So one of his most famous things, he was interested in how do fibers in a bundle work together? How do they interact? And he did all the mathematics and figured out the physics and stuff, and then he wrote the paper. But being Ray, he didn't actually write about bundles doing it, but he actually wrote the paper to explain why ponytails have the shape that they do. And for this, he won the 2012 Ig Nobel Prize for this, of figuring out that. But other things that he's pondered and written papers about, so he's got a whole career about this, have been things like, why are icicles shaped the way they are? Another question he asked, our clothes are actually made up of really tiny fibers. And so he's addressed the very important question, I guess if you're modest, why don't our clothes fall apart? Why do these tiny little fibers allow us to have cloth and clothes? And more recently, this last summer, we had a lot of release of cicadas that come up every 17 years in the United States. He wasn't interested in the 17 years, but he was really interested in how is it that they all know to come out at the same time? And he looked at that. Actually, he and his wife, who is Adriana, who's a wonderful mathematician, and they figured out that it can't be random, that there had to be some sort of communication among the cicadas, probably underground, where they told each other it's time to come out, which is probably good for mating, to be able to have others there. But he's done all these things, problems that are facing all of us, or that all of us could see. He's made a career of this. And I want to tell you one more example, because it is from a high school student. I've been very privileged to go every year, or almost every year, to what's called the International Science and Engineering Fair. It's the culminating high school science fair for people from all over the world. They usually have now I think it's about 2,000 students from 70 or more countries. And one day I was walking through the displays, and there was this young woman at her posters, and it was a little strange, so I stopped and I asked her about her research, and she said, probably the strangest introduction I've ever heard to a scientific talk. She said, well, the first thing you should know about me, I'm a ballerina, and I'm actually a very good ballerina. Well, I wanted to hear what the rest of the talk was about. And what she did, her name is Abigail Fried, she talked about a problem that the very top echelon of ballerinas have with their shoes. And the problem is that the shoes have a cardboard sole. And so after a single performance, that cardboard sole has lost its flexibility, and they can't use the shoes anymore. So it's $110 or more every time they do a performance, and you need a new pair of shoes. It becomes a very expensive art form to do this. And so what was her project? Her project was to come up with a better sole for the shoe. And she found a carbon composite material, and she tried different layers of it, counting them, and she found that one on the left here was much too flexible, 3 turned out to be perfect, and 5 was much too stiff. And she then reported, she told me that the shoes keep their flexibility, not for one performance, but for months. And I thought, this person should win the prize here. I'm not a judge, so it wasn't something. But it was just fantastic that she came up with her own problem, realized it, and then did this. So let me go from there to talk about this idea of questioning assumptions. And the examples I like to give, Peter said, I like puzzles, I do like puzzles, and there's a type of puzzle that if I were lying to you, I'd say I only liked when I was a kid, but in fact, I still like them, which are called matchstick puzzles. And what these are is a puzzle. Here's one example. Here's a pattern of matchsticks. Can you move the matchsticks? Can you move only 3 of them so that you wind up with 3 squares? And so you sit there and you swear at it, you manipulate it around, and you play with it. And eventually you come up that, yeah, you can. You can come up with 3 squares. And then you look for the next one. And this website will get you to a whole series of these things and solutions. But here's another one. Here's a fish pointing in one direction. Can you get the fish to go in the other direction? And so you maneuver things around, again, swearing a bit, until you finally find that that's the way to do it. And that's all fine, because you learn how to do something, and you just apply it more. You got to move things, so you just move them around. But then you get this problem. Take these 6 matchsticks and make 4 equilateral triangles. And that's a little hard to do. It's sort of impossible until you start questioning your assumptions. And so one of the assumptions that all those other problems had was that they were done in 2 dimensions. If you throw out that assumption and say, well, let's use 3 dimensions, then you get that pattern. And suddenly you can make 4 equilateral triangles with the 6 matches. That's not the only assumption in this problem or the only solution, because there's another solution. All those other things made people think, well, they had to be the same size. Nothing said that they were the same size. Why should they be? If you throw out that assumption, now you can have 2 large equilateral triangles and 2 small equilateral triangles, and you can get an almost planar example of this. And then after you realize that you have to throw out all your assumptions, and I'm going to ask for hands but not answers for this one, you get this problem. Make these 3 matches into 6. You cannot break them in half, okay? If you know how to do that, raise your hand. A couple people have done that, okay? There was someone in the back there that raised their hand. Say the woman in the white blouse. Yell out what you think the answer is. Absolutely. Roman numerals. Okay? It's the assumptions, right? It's what was the problem, what was it really asking, and what do you do? I have a good friend who is a spectacular neuroscientist, Mu-Ming Pu. And one day, a bunch of us were sitting around, sort of in awe of this guy, and said, Mu-Ming, where do you get all your ideas? They're incredible. You have come up with all these wonderful ideas. How do you do it? And he said, I use the retrospective approach. We didn't know what the hell he was talking about. And he said, well, look, I read a textbook. I'm not fancy papers. I read a textbook. And for him, the textbook was the 2nd edition of Molecular Biology of the Cell. And he says, I read what they say about nerve cells, and I ask myself the question, how do they know that? Why do we say that? And he usually finds out that they're based on rather old experiments, rather bad methods, things that were extrapolated from the results but not really proved. And so he uses more modern methods and retests the hypothesis. If it's right, he now has strong evidence to confirm it that's a nice paper. If it's wrong, he's got new information to refute the old ideas. And he just did this repeatedly with wonderful, wonderful results. And then, as I say, publishes the correction. I want to finish. So these are all ways I think we can approach science, approach discovery while we do this. I would also add one other thing, and that was the thing about taking risks. Oliver Smithies, another Nobel laureate, but he died a few years ago, used to talk about weekend experiments. Weekend experiments are experiments you do on the weekend because you're too embarrassed to tell anyone about it. You know that on the weekend no one's going to be there, but it's just killing you to find out. So you do the experiment Friday night, Saturday night, sometime when no one else is around, and if it works, you crow about it on Monday morning, tell people and if it doesn't work, you just ask people how their weekend went. But it's that why not try it? See if it works. If it works, terrific. So anyway, I want to leave you with two quotes. The first is by Robert Wilson. This is a different Robert Wilson. He was the first head of the particle accelerator called the Fermi Lab. This was the biggest particle accelerator in the world until the CERN Super Collider was produced. And in 1969, he was asked to talk to a committee of the U.S. Congress to explain why the U.S. government should put all this money into making this particle accelerator. Now, 1969 was the time that the war in Vietnam was taking place, and so there was a great concern about national security. And so one of the senators asked him, how will this help? Tell us all the ways this will help the national security. The senator actually was trying to give him a softball question that all the other senators and members of Congress could get behind. But he gave the answer that the senator didn't expect. He said, oh, how many? None. And so they asked him the question a couple more times, and finally he said, no, really, in what respect will this particle accelerator help the national defense? At which point Wilson said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with, are we good painters, good sculptors, great poets? I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. My final quote is from my daughter. This is my daughter in 2004 when she was 12 years old. And I think she found sort of the secret of why basic research is so rewarding. In 2004, I was elected to the National Academy of Sciences, and my wife and daughter and I went down for the induction ceremony. The induction ceremony is the most boring ceremony in the universe because there's about 70 people, one at a time, get on the stage, shake hands with the president of the academy, have a picture taken with the president of the academy, and they walk across the stage to a book. Now, they're told every former member of the academy has signed that book. So you get really excited because you think you're going to sign the book that Einstein signed. Except then they tell you, no, I actually started the book in 1970. So you get real. But as you walk across the stage, there's somebody on a microphone that reads the one sentence of why you got elected, what you did for them to consider you for membership in the National Academy of Sciences. Now, my daughter, at the time, she's 12 years old, perhaps not the person with the greatest amount of patience in the world. And so I eventually made my way down, and I said to her, Sweetie, you know, I'm really happy that you and Mom are here because to help me celebrate, but I have to tell you, you must be bored out of your mind. So I just want to apologize. Thank you, but apologize. And my daughter looked at me, as she has often looked at me, like I am the stupidest person on the planet, and said, "Dad, don't you understand about these people?" She had been listening to all those sentences. And so I said, "What do you mean? What about these people?" And so she said, "Dad, they were the first people in the world to see or do what they did." I think that's astonishingly astute. It is the reward we get for all the failures, for all the missteps. We get to be the first people in the world to see something or to do something, and that's very rewarding. Thank you very much.
That was astonishing. Thank you very much, Marty, for this unbelievably inspiring presentation and your thoughts about science. So now the stage is for the audience, for the students. So please ask questions. This is the time for you. So please raise your hand.
Yes, go ahead. Yeah, you. Oh, you got to get a mic.
All right, so first of all, thank you for this nice presentation. I actually have two questions. The first one is, as far as I understood your presentation, it was about thinking outside of the box. And my question is that, is it possible to think outside of the box if we don't know what the box is actually?
So that's a great question. You know, I never know when I'm going to be inspired about things. People have sometimes asked me, it's going to sound like it's an unrelated question, but it really isn't. What was the important thing in your training that you think helped you being a scientist? And so how do you, what in my training helped me, I think, to think about things differently or combine it? And it was when I was in college, it was the humanities and social science courses. Because what we did in those courses is we read a lot of material, a lot of articles, a lot of books. We learned a subject, and then they'd have a final exam that we had never seen the question before. It was completely different. And what we were supposed to do is grab whatever knowledge we remembered, whatever we had learned from the course, and synthesize a completely new answer. We had to make something up. And I think that training was spectacular, because that's what we do as scientists. It's not always the straightforward things. That's always good. But it's having a problem in the back of your head and then suddenly seeing something where you have a connection. GFP was something like that for me, because I was working on a transparent animal and I was looking at gene expression, and I suddenly hear that there's this molecule that would allow me to look at that in the transparent animal. So it's taking something from here and something from there and something from somewhere else and somehow synthesizing those things. And so for me, it was those humanities and social science final exams that I probably complained a lot about, but I think I learned how to sort of synthesize material from. At least that's my thought. So I hope that was it. You said you had a second question?
Yes, my second question is that this kind of thinking outside of the box is tautable, or it's some kind of natural gift we're born with?
So I had hoped that what I was saying in the last part of the talk were some ideas about how to train ourselves, to ask questions about what we see around us, to ask questions about what are we assuming here. So it really, so to me, those are two approaches of thinking how, am I thinking about this in a conventional way? Is there, maybe that's the right way to think about it, but is there a different way of thinking about it? What am I assuming here? I'll give you another example of something that you've seen every day, but I'm pretty sure no one in this audience has actually, well, maybe one person, has actually asked themselves any question about that. You know, there's an old experiment, I think it was old when Darwin repeated it, of taking a plant, you put a seed in a pot, you grow a plant, and the root goes down and the shoot goes up. And you take the plant, you put it on its side, and now the shoot goes up still, and the root goes down and curves because they're responding to gravity. And people have investigated that. And that's as far as usually most textbooks go. But if you go and you look at trees, which we all have, what you find out is that the branches on the tree also respond to gravity in a very species-specific way. So you get fir trees that can be completely parallel to the ground, all their branches. You get some other trees in which they're sort of pointed up at an angle. You have trees where they point straight up, like the Italian cypress tree, that all the branches go straight up. And you have some trees that appear to be mutants because they don't respond to gravity at all, like weeping willows, where everything droops. And so all of this is called a set point in plants, and it's something that people have been studying, but we've all seen it and not recognized it. As you leave here tonight, especially now that the leaves are off the trees, you're going to see that the leaves, that the trees have a characteristic pattern to them. It's another one of those questions that's right in front of us. Anyway, other questions, or have I gone through the time? There's somebody way in the back there.
I would like to ask for a suggestion, actually, and if we're doing a research and somewhere in the procedure we get stuck or have a, I don't know, burnout or something like that, what would be the best thing to do?
I've been asked this question a lot, and I give the world's worst answer to it. And I'll give you the example from my own work in the lab. From my postdoc on, I have been really lucky to look at a set of mutants in the nematode Cenorhabditis elegans that are defective in touch. And we've found many genes that when they're mutated, the animals become touch insensitive. You might ask, how do you test touch in an animal that's 1 millimeter long? You take an eyebrow, you glue it to a toothpick, and you tickle the animals. If they're touch sensitive, they move away from the tickle. If they're not, they don't. Dead animals also don't move, so you need to differentiate them. But we have found many genes that can be mutated so the animals are touch insensitive. Some of those genes result in touch insensitivity because the gene that's been mutated is needed to make the sensing cell. If you can't make the cell, it won't work. You won't be able to sense it. So these allow us to address a question of cell differentiation. Others, the cells are made. They just don't work. And those don't ask a developmental question. Those mutants allow us to ask questions about what are the molecular mechanisms that sense touch. And we've done both areas of research, and we do them at the same time. Sometimes one works, and sometimes the other works. So the idea that I get from that is, if you are lucky enough to work on more than one thing, if one is not working, let it simmer and work on something else. This is equivalent to saying do twice as much work, and I know that's not a good answer. But it really is something like that. If you look in a laboratory, you will see that maybe individual researchers in the laboratory working on their project are unhappy. But the head of the lab is usually ecstatic because somebody's experiments are working. So this multiple approach, I think, is one way of dealing with that. It's very hard because when things don't work, the last thing you want to do is more work or to go back into the lab. But I find that if people have multiple things to think about, something usually works out.
Thank you so
I don't know if that was useful or not.
Other questions? Somebody's...
Yeah.
Okay. This question is kind of similar to what she said, or she asked. And it's when I think of an experiment, like the last time that my teacher assigned us to have a hypothesis and then, of course, come to a conclusion, I struggle with the fact that I want to know way too much. I have an idea, and I have no idea how to test it, or I have no idea where to start it, like to take it into small, well, you could say bite-sized pieces. Would you have any advice for that?
So I have two reactions to it. The first is talk with people, diagram things out, interact with people about it, because the more interaction, the more input you can get. It's not you tested against the world, it's you're trying to solve a problem. So that you should know you're not alone, that there are other people to interact with and talk with. The other thing has to do with hypotheses. You know, the standard scientific story is that some genius scientist wondered about a problem, came up with a hypothesis, dreamt up the perfect experiment to test the hypothesis. It worked the first time, and they got all sorts of prizes. That's the story, that the hypothesis, the problem is there, the hypothesis comes first. But what I tried to say in my talk is that a surprising amount of the time, while you're working on whatever hypothesis you're working about, something unusual happens. And so the discovery, I think, comes before the hypothesis, not after the hypothesis. Because, to cite Fermi again, after the hypothesis, you made a measurement. Before, if it's not what you're thinking about, it's something unusual. If it's those rats that are suddenly not thriving or thriving when they're with their mothers, that's something unusual to look at. So I think that it's a little bit different. You do an experiment, for a good reason, hopefully, but as you do it, something unusual comes up, and you make the discovery. Once you've made the discovery, then there's a whole bunch of questions to ask, and so that's when the hypothesis comes in. So I don't think it's hypothesis first, then make the discovery. I think it's the discovery and then make the hypothesis.
Thank you very much.
Okay, and the last question, due to time,
Yep.
maybe. Yeah, thank you.
Thank you very much. So my question is, have you ever tried to put GFP into plants, or would it worth it?
So people have put GFP in plants. One of the first things that was done, a scientist in Cambridge that put it. And sort of at the beginning of GFP, when people were starting to do things, I would periodically get telephone calls into my office, and they were always of a couple of types. One was, could we put GFP into plants and put them all along the roadside so that the headlights on the cars could activate them, so we didn't need to have lamps on our thing. And I don't think anyone actually produced that, but that was one of the things. And the other thing and maybe this is more for just the United States, always in sort of February or so, someone would call me up and say, look, St. Patrick's Day is coming in March. Is there a way of making green beer? That we can make green fluorescent beer. And I think somebody actually wanted to make green fluorescent chewing gum. Anyway, so yeah, there's been people, but people have definitely put it in plants.
Thank you very much.
Okay, thank you very much, Marty, once again, and please stay there because we also have a certificate for you. And also, we also very much admire your background concerning STEM. So in Hungary, we made a stamp for Marty for the appreciation of being here.